So when you look at this picture here of these insects, all of these insects do share something in common that we refer to them as beetles. And when it comes to beetles, there are a great number of species that have been identified by science, for over 100,000 of them, and over 300,000 actually. And as far as insects go, they're one of the most abundant, or the most abundant animals that at least they have been identified and known to science. And so the question is, how do we get a handle on this? And that's what this chapter is about. How do we get a handle on all of this diversity and the number of organisms that we see on the planet? And that's where this chapter is going to help us provide a context in which we're going to study and survey the different types of organisms that we see. We're going to be studying plants and bacteria and animals and fungi and so on. And so that's where we're going to be using what's called systematics, which is the study or the science of trying to figure out how these organisms are related to each other, and the hypothesis for doing that, or that are developed from those studies, are referred to as phylogenies. So we're going to be focusing on that, and also how to name these organisms. Comparative biology is part of that process, and we'll be doing some of those comparisons as we progress. So we're going to go ahead and skip that part of the chapter. And as we progress through our survey, we're going to be doing some of those comparisons anyway. So our first section is called systematics, and one of the first objectives here is to be able to understand what the word phylogeny means and apply it. The phylogeny is basically just the relationships between organisms. It's a proposal based on evidence. And then we also want to see that back then and even today, the way we try to establish is based on similarities. And prior to that, it was looking at just physical appearance. And nowadays, with molecular biology, looking at DNA, molecules, and proteins, we also use those lines of evidence, fossil evidence as well, to try to establish those relationships. But sometimes these similarities can be misleading. And a great example would be when you hear someone, and it happens from time to time, call a whale a fish. And whales are not fish, they're mammals. And so mammals are grouped together for a reason, because they're related to each other by evolution. And so we're going to talk about that. So we want to start by looking at, first of all, that all organisms do share some of the many characteristics that we see. For example, we're all made of cells, humans, bacteria. The individual unit of life is a cell. We all have metabolism. That means we're breaking down and building up molecules. We also use ATP as a sort of universal energy source for doing the work of life in the cell. And all life uses DNA as genetic information for running the show, and how the organisms develop and running their metabolism and so on. There is quite a bit of diversity of life here, from the most microscopic and somewhat simpler in structure bacteria to some of the largest organisms multicellular, including whales, and these giant sequoia trees, which are a type of conifer, cone-bearing trees that we'll study later. So again, what we're going to be using here is we're going to be looking at characteristics that are shared within these groups, and that sharing of those characteristics might imply their evolutionary relationships. So we look at evidence and comparisons of organisms that are alive today, but we also look at fossil records. When you look at fossil records, they're not always complete, so that makes the job even more challenging. But we also use other types of evidence, like I said, molecular approaches, where you study DNA sequences and so on. And basically, we're going to use those to develop hypotheses about the relationships. When we say relationships, we're looking under the umbrella of evolution, biological evolution. So what are their evolutionary relationships? We go back far enough in time, there was no life on this planet. As you progress towards the future, life evolves, and then as life evolves, it goes through changes. Okay, so and those changes, as they progress, leave behind clues in the fossil record and in organisms today that help us try to understand those relationships. So this study is referred to, in establishing those relationships, referred to as systematics. This is a science, so it's an area of biology where you try to reconstruct those relationships. And once that those relationships are constructed, they're subject to testing. These are hypotheses. They're not proven, and new evidence may change the way we view. And that happens all the time. That's the way science goes. All right, so a phylogeny is the hypothesis of that evolutionary history. So you want to be able to use that term correctly. Phylogeny is the pattern that we see in the relationships in these organisms, their evolutionary history, and systematics is the science of doing or establishing that. Now, Charles Darwin viewed these types of patterns in his travels around the world, in his studies of domesticated animals, selective breeding. He knew that from this evidence that organisms do go through changes over time, looking at fossils and so on. And so he viewed life on this planet as a branching tree. And if you went back far enough in time, you could trace species back to a single common ancestor. And so these are his notes down here. And you can see his little diagram of a tree here. And at every node, we're going to be using that term later, every branching point is their node. All of those organisms that come off of there, in this case, in his drawing here, all of those branching that are all descended from this common ancestor back on that node. So that node would be a hypothesized common ancestor. And they refer to this as descend with modification, just like we use descendants today, like I'm a descendant of my grandparents, whereas my grandparents are my ancestor. So we kind of use those terms, but in a context of greater periods of time, on the order of millions, even billions of years of time on the planet. So when we look at these proposals, a lot of times of how we think the organism is based on evidence, how we think organisms are related to each other, a lot of times we draw these branching trees. And this is one of those kinds of drawings. It's called a cladogram, and it's referred to as a cladogram because it uses techniques in a technique called cladistics. But it's basically, these are phylogenies or a proposed phylogeny. And my question to you is, I want you to hit pause to study these and these three different diagrams and see if there's actually a difference or not. And so go ahead and do that. Look at it real quick. Press pause and then look at it. I'm going to go ahead and give you the answer right now. But if you press pause and try to look at it, you can compare it to what I'm going to tell you right now. The answer is all three of them show the exact same phylogeny. And if you look at it, and the key here to interpreting it is to look at the branching points. And if you start at the top of the diagram where all of your species are listed up here, okay, and you go down the diagram, it's like you're going back in time. Remember, in Darwin's view, descent with modification, you start with a node further back in time, and then as you go back down the branch towards the ends, you're branching off, and at every node is a common ancestor. And so here we can see that if I identify the first branching point going back in time in this diagram for all these organisms that are related, the first branching point is the human and the chimpanzee. And if I come and look over here, I see the same thing here, and then I see the same thing here. And you can continue to do this. The second node down here shows that the human and the chimp are more closely related to the gorilla, and they share a common ancestor, but you have to go back further in time. And so the human, chimp, and the gorilla share a common ancestor, but you have to go back further. And they're more closely related than those three are to the orangutan. You have to go back further in time to a node there, and so on. And if you studied all three of these, you can see that all they really did was just spin these things around one of those branching point axis, around an axis on that branching point. If I were to rotate right there where I just drew right here, you're going to get this diagram here. If I were to rotate this particular diagram and then turn it sideways, and I rotate it on the lower branching point here, just flipped it around there, you're going to get this diagram. And they all still have the same nodes and same connections or same relationships. So you see this sort of a nesting that goes on in there. So again, the key here is to look for similarities to help establish these relationships, but that can present some problems. So the idea here is it's very easy, and oftentimes it's done, is to have the assumption that evolutionary rates are are going to be constant, and so in other words, the pace at which changes occur, that's not always the case, and that can lead to problems in trying to determine at what point we have these branchings, and so that can be a problem. And then sometimes evolution isn't always in one direction. Sometimes a characteristic evolves, something new appears in an organism that we didn't see in fossils that were older, and then all of a sudden it disappears again, and so that would be a reversal. So evolution isn't always in one direction. And an example given is the evolution of horses. There's a tendency for, if you look at the fossil record for the horses, the ancestry of horses, those fossils, you start to see a progression of bigger and bigger, but some branches show them going smaller, so there's a reversal there. Another problem is convergent evolution. And convergent is the opposite of how we view a tree. A tree is always branching. And so your ancestry here gives rise to these divergence in these characteristics, this separation of these characteristics. But sometimes two organisms on two different, or two groups of organisms on two different trees end up evolving a similar characteristic. So that can present some confusion. And this is where some individuals might call, not without knowing, when they look at a dolphin or a whale, they might call it a fish. It's less common now because there's a lot of great nature shows on TV, but in those cases, when you have two organisms, two different branches, that would be called convergent evolution. Now, I kind of got this diagram here to show you. Now, all of these organisms that are here all show this sort of streamlining characteristic, and that's good for moving through the water. Okay, and they all have this backbone within the body, just like we do. In fact, we share that characteristic with them, and we go back far enough in time we have that characteristic. But these are not all closely related. The dolphin is a mammal, and mammals are on a different branch than sharks are. In fact, fish evolved first than mammals did, according to the fossil record. And then we have a group of birds called penguins, and then this is the ichthyosaur is actually basically a group of extinct reptiles, right? So we all have this. They all have this common characteristic of the streamline, because they're predators and they go through the water. They have to be able to go through the water, but their ancestors didn't have this fine streamlining. Okay? Now these fast predatory fish already had that, but some fish moved on to land. They evolved legs. And some became reptiles, others went on another line or another branch and became mammals, and some became birds. Okay. In fact, this dolphin's ancestors actually had 4 legs, and these legs had been lost. In fact, some whales still have these hip bone inside that shows evidence that their ancestors had legs, because the hip bone connects the legs there. But what happens here is, as these groups of mammals start spending more time in the water, some of them are just going to have a better shape for moving in the water and may be able to take advantage of being in that water more efficiently, swimming after or finding food, for example. And if you can do that, you're going to survive better. You're going to have more energy available in your body because you ate more for reproducing, and then over time you're going to be leaving more offspring than those that don't have that shape. So over time there's this pressure for having a better shape for moving through water, and that would be convergent evolution, when you see a mammal start to have the same body shape as a shark, or a reptile, group of reptiles called ichthyosaurs have that shape, or a bird like a penguin. And so this diagram here highlights that. Here's a note here where we have the evolution of vertebrates, all animals with backbones, and so we have a divergence. We have the fish that evolved early on, and then you have vertebrates that move on to land. We call them tetrapods, and some of those became mammals. And so here we have an ancestor for all mammals, and then there's a divergence, and you end up with some that went off to live in water, and those that are living on land, these carnivores. There's many groups of mammals, right? So we can draw a lot more branches, but the group that went into water lost their legs, and it started to look more and more fusiform. And so while the dolphin and the puma here actually share a more recent common ancestor, we might be tempted to call the dolphin a fish when it is not. So here The streamlining would be a shared characteristic that can be deceiving and saying, look, they're related, closely related, but they're not. So that would be an example of convergent evolution, where you're on two different branches, and then these two organisms independently have this evolved this similar character. So that would be convergent evolution. So that answers that last question on how this phenotype, this characteristic, the way it looks, can be misleading. So in section 2, it's titled Cladistics, and cladistics is a method for or technique for determining phylogenies or evolutionary relationships among organisms. And so the learning outcomes for this section include for the learner to be able to distinguish between what's an ancestral and a derived similarity or character. And then we should be able to explain how characteristics that are shared within a group are the ones that are important in determining close evolutionary relationships. And then also be able to interpret and even produce a simple cladogram that shows given data that shows those evolutionary relationships. So looking at cladistics as a technique for systematists to determine phylogenies or evolutionary relationships, one of the things that has to be done is to recognize the difference between ancestral and derived characters. So an ancestral characteristic may be seen in a broad group of organisms, and that characteristic was something that was inherited from some common ancestor. Whereas a derived characteristic is something, a characteristic that has evolved in a subset of that broader group. So something new that's seen that would not be seen in all members of that larger group. For example, we're going to be talking about vertebrates soon, animals with backbones, and all animals with backbones got the backbone from a common ancestor. But then when we go further, as descent with modification is occurring through evolutionary time, we have hair evolving in one group or on one branch, and that would be mammals. And so here, the ancestral character would be the backbone, whereas the derived character would be hair, only seen in a subset or subgroup subgroup of that larger group that you're studying. In cladistics, guys, only shared derived characters are the ones important that show these close evolutionary relationships. So cladistical methods will use any kind of characteristic that are available to them. And these characteristics include morphology, the structure of the organism, behaviors. A lot of times behaviors are inherited from their ancestry. And so behavioral characteristics, physiology, this is the internal functioning and workings, even at the cellular level, the biochemistry that's going on. And then even DNA sequences can be used, comparing DNA sequences for shared characteristics. So in order to use these as characteristics, we must be able to identify, first of all, the character, and then the character has two states. One would be an ancestral state, and another one might be a derived state. And so the idea is to define that. And so when we look at vertebrates, for example, tail might be a character we're looking at, and the presence of a tail in vertebrates was actually inherited from a very distant ancestor, so that would be ancestral. And then a loss of tail, like we see in humans and apes, and even frogs, would be something new and derived within those subgroups. So this is the idea here, is you would get a bunch of characteristics relevant to the group you're studying, and then try to determine whether or not these characteristics are derived or whether or not they are ancestral. And so you can see the top of the table has some of these characters that we are going to be examining, including jaws, lungs, amniotic membrane, whether or not you have hair, and no tail. So no tail is something new, because an ancestral, we know from other studies, is an ancestral form. And then bipedal, walking on two legs. And then you have a bunch of vertebrates here, in this case, that we'll be studying as a larger group. Lamprey, shark, salamander, lizard, tiger, gorilla, and human. And here we go through and we rate as either a 0, which would be ancestral, or in other words, not having the derived character, and 1 if you have the derived character, as the key indicates down here. So you can see that the lamprey has none of those. The lamprey doesn't even have jaws. They're called jawless fish. And then the shark has the jaw. And then if we come down to tiger, the tiger has a jaw, lungs, amniotic membrane, and hair. Okay. And so once you do all of that, you can use that data to produce a diagram that shows the phylogeny. In this case, the specific diagram, since it was produced using cladistical methods, would be called a cladogram. And so here's what the cladogram would look like based on the data from the prior example. And in this diagram, it really helps you see what would be derived and what would be ancestral here. So if we look quickly here, the way we'd interpret it is from the last section, at every branching point or node, for example there, all the organisms are coming from an ancestor, a common ancestor. So your node would be your common ancestor, a hypothetical one. We don't really know what the common ancestor is, especially because the fossil records are not complete. And from there you have a divergence. So you have a group that becomes a lampreys, and then the group that evolves a jaw. So here's your first derived character jaw, and that defines anything above there. So now if we look and specifically focus on a group up here that we would call mammals. And if we go back down the tree and we look at that node there would be a common ancestor to this mammal group, and that shared derived character that evolved that we do not see in others would be hair. So in this case, hair would be the derived character for mammals. And then if we looked at something like the lungs would be ancestral. The lungs evolved earlier here when these animals moved on to land. And that group actually is called the tetrapods. So while the gorilla, the human, and the tiger have lungs, that doesn't mean that makes them more very closely related to the salamander that also has lungs. They're more distantly related because lungs is a ancestral character. You have to go back further in time. So as you go down this diagram, you're going further back in time to some more and more distant common ancestors. So we can interpret this diagram here to show that the closest relative to the human is the gorilla, right? And it would be the human would be more distantly related to the salamander, and the relationship there would be the lungs that were inherited from some common ancestor way back. So a lot of times when we put these or do these drawings based on analysis of these characters that we're rating for the presence or not having the presence of these characters, we also want to compare them to an outgroup. And so the outgroup is not going to have any of those derived characters that we're analyzing. In this case, your outgroups, they're usually put off to the side there, and they're going to be at the lowest node on your diagram here, going back the furthest amount of time. Your outgroup here would be your lamprey, and the lamprey is there for comparison with these derived characters there. So the first step, though, in doing this analysis is you want to have some characters that can be analyzed that are relevant to the group. We've already talked about those for, in this example, the jaws all the way to being having no tail and being bipedal. And we want to polarize them. And so polarizing is what we're going to determine whether these are going to be ancestral or derived in their nature. And so, for example, we might polarize tail and whether or not that's present or absent. Now, tail is not even on the list of characters there at the top, but tail is something that all vertebrates have. These are animals with backbones to begin with, so we could actually add tail here. In fact, the lamprey in the outgroup here has a tail as well. The tail is defined as this extension that extends beyond where the anus or the exit for the digestive system, where waste is left out. So again, the outgroup there would be the lamprey. Doesn't have the derived characters that we're studying, but where would tail be? we could continue to extend this cladogram further outward, and if we did, we would have another closely related group that doesn't even have vertebrae. They're called lancelets. They're little invertebrates that do have a tail, and they do show a close kinship to the rest. You have to go back further in time. And those characteristics that are shared by them would be discussed later on in the course. But if we look at this node right here, one thing that's shared by everything from this ancestor that evolved would have been the backbone. But long before the backbone evolved, there was a tail. So your tail is actually an ancestral character that all of them do possess. And we also see that in your outgroup. So the outgroup for the lancelet has the tail, right? Even the lamprey, the original outgroup we were looking at, has a backbone, but it has a tail as well. Okay, so again, the outgroup is for comparison, and then the rest that are here, we're looking at those derived characters that would have evolved. And so when you do that, you end up with a data table that looks like this with ones and zeros. Zeros meaning the ancestral form is still present, and then the derived form. So for example, the amniotic membrane, which helps protect the embryo from drying out. Frogs don't have this. This is why frogs need to reproduce in the water. The eggs need to be there. But then the amniotic membrane evolved. That's derived. So we don't see that until we see lizards. The salamander, amphibian, the shark is a fish, lamprey, those are tied to water. They don't have the amniotic membrane, so they're assigned zeros. Ones for those that do have it. So when you look at these comparisons, you're looking at groups, say a shark group or lizard, tiger, whatever group you're looking at. When you're looking at a group and it displays or it shows multiple character states, and so we're multiple characters, and they're displaying these, and one of those characters they have is also seen in the common ancestor. Okay, so the one that's seen in the ancestor would be considered to be an ancestral character. So an example already was given was with the tail, right? So the tail would be that ancestor. Now it says that it's going to be most reliable, this ancestral character, if it's seen in other outgroups. So right now we have the outgroup of the lamprey, but earlier I drew an even more distant outgroup we could have compared to. Let's say we wanted to compare even looking at the lamprey and include the lamprey in our actual study group, not as the outgroup. We would see tail is present actually further in even more distant ancestry there with lancelets that we drew earlier. So what does this mean then? So you start looking at all of this here. All of these organisms had tails, and that means when you run across an organism that has all these other derived characters, the amniotic membrane, the hair, lungs, and so on, and then you see no tail, but it has all those other characters of hair, amniotic membrane, lungs, what you end up seeing is that tail loss then is not an ancestral but a derived character that we see within gorillas and humans. So when you're going to do this type of analysis, you're going to ultimately construct this cladogram, which is a picture that shows their evolutionary relationships using that polarization of those characters. What you end up producing is a diagram that shows evolutionary units, which we're going to call clades. And a clade then is defined as a group of organisms that shares common ancestry. And what you really get is clades nested within larger clades. For example, here we have the tail loss clade. Okay. And then a clade you're probably familiar with is mammals. And so the tail loss clade is defined by losing the tail there with some common ancestor that lost the tail, and then descent with modification, you get the different groups with no tail. When we look at the mammal clade, which is a broader clade, it shares common ancestry with gorilla and the human, and we have a node right there, and the derived character would be hair. So each clade is going to be defined by an ancestor that has evolved this shared derived character. And that evolved, or that new character that arises within these groups. For example, you're going to move on land, you're going to need an amniotic membrane, so you're an amniote. that broader clade is defined by that derived character. So if we're studying amniotes, right, your salamander might be your outgroup to compare for the amniotes as we study in amniotes. At any point, though, at any node, that characteristic that is shared by everything above that node in your diagram is going to be referred to in cladistics as a synapomorphy. So basically, what is a synapomorphy in cladistics? It's a shared derived character. It's when you're talking in cladistics, you just call it a synapomorphy. That's a shared derived character. Okay, it's your derived character that is shared. Okay, so again, we're going to be looking at many groups of organisms, and we want to be thinking, what are their relationships to other organisms? And in that sense, we're going to be referring to clades, these evolutionary units that share that common ancestry. Okay, so when you're constructing these cladograms, some careful consideration has to be given to these characters that you're studying. Especially when it comes to plesiomorphies or plesiomorphies that are characteristics that are ancestral. Sometimes those ancestral characteristics through the process of evolution disappear on you and we don't see them anymore. They evolve away. In other cases, we see that ancestral character in the broader group. For example, tail. Okay, and tail would be this ancestral character that's shared. And so an ancestral character that's shared is called a simpliciomorphy. And you have to be careful because although it's shared, that shared ancestral character doesn't imply a close relationship. Okay. An example here, if we were just to look at tail alone, and you looked at lizard, and you say, well, a lizard has a tail and a tiger has a tail. And you say, well, those two must be more closely related than the tiger is to the gorilla. And that's not the case. It turns out that the tail is an ancestral character that's shared by the broader group. Okay, but the tiger is more closely related to the gorilla and the human because they share something that evolved later than the tail, the hair, and the lizard is diverged before that hair evolved there. So the lizard diverged here when amniotes first evolved, and the lizards went down their own path of evolution. Hair evolves later in your mammals, and so just because you have a tail, you're not closely related. The tiger is more closely related to those tailless mammals called apes and humans. Okay, so we want to be careful with simpliciomorphies, which are shared ancestral characters. We don't want to confuse them for implying a shared, a close evolutionary relationship, which is what talked about right there. So some things that can complicate further in cladistical analysis would be the idea of homoplasy, which is another term. There are already a lot of terms being thrown here, so I want to make sure you interact with these terms and practice using them, apply them in the right instances. So I do have some basic review questions that you can look through or make your own questions about this material here. But what is a homoplasy? This is a shared character state that in this case could be confused for showing a close evolutionary relationship and it'll show common ancestry for that character, but it was not the case. In this case, this might be due to convergent evolution, like the example I gave earlier where you have dolphins, which are mammals and are on their own branch, ending up looking like the shape of a shark, which is very streamlined for moving very swiftly through water. That's a case of convergence, and when that occurs and the character that you're looking at, say, is a streamlined shape, that character would match those two together closely, but they don't share that characteristic because of a common ancestor, so that would be considered a homoplasy. Okay? Now that occurs through process of convergent evolution, and one mechanism for convergent evolution might be a reversal. For example, we have groups of animals that have evolved a hollow fluid-filled cavity, a body cavity, and then some of them lose it, okay? And they don't have a body cavity, and so they lose it later. whereas other groups of animals diverged even before the body cavity evolved and never had a body cavity to begin with. So here you have one group on one branch that never had a body cavity, and they're still alive today. They just never evolved. They branched off early on. And then another group, the body cavity evolves on that other branch. And then as evolution is occurring, then some of those branches, finer branches towards the end, they start losing that body cavity and start to look like the ones that never had a body cavity to begin with. That would be a reversal that results in convergence. Those create homoplasies. Okay. So what do you do in cases where homoplasies occur and could present confusion in the hypothesis you want to go with when it comes to establishing your phylogeny, your evolutionary history? You're going to apply what's called the principle of parsimony or parsimony. And this one here is you're going to go with the hypothesis or the phylogeny that requires the fewest assumptions. And this is illustrated here with the idea of tail loss, because frogs don't have a tail either, but frogs are evolved in a group in which their distant ancestor had a tail. Okay, so the frogs lost their tail, it's a derived character, and humans and gorillas lost their tail, right? So we could actually propose two different phylogenies that's displayed here in these two cladograms here. Remember, cladistics is just a method for trying to uncover a phylogeny, right? Hypothesize a phylogeny. And when we look here, and we look at the changes that must occur here, these changes we could analyze in terms of derived characters, a new evolutionary change. Here, you have amniotic membrane here, tail loss, and then tail loss again over here. Okay. Now, tail loss is common to humans and frogs, so we might be tempted to put them together, but we put them on separate branches. And in this hypothesized phylogeny, there's only 4 changes that occurred, 4 evolutionary changes. If we try to put the frog together with the other tailless ones, because we think that's a better fit, then we see that 5 changes are needed. Amniotic membrane evolves, then hair, then the tail loss, then we have to lose the amniotic membrane, a reversal, and another reversal of losing the hair. So with 5 changes, it seems that this hypothesis would be better because it follows that principle of parsimony here. And in this case, it is homoplasy that would have caused us to have a hypothesis that may not actually reflect evolutionary history here. It seems the better explanation is the frog and the human group, the two evolved to not have a tail independent of each other. They were on different branches. They didn't get that tail loss from the same ancestor. They just happened to evolve that same characteristic independent of their ancestry. Okay. So biologists are now using more and more DNA sequences to help derive cladograms, which are hypotheses about their phylogenies, right? And so here they're going to use segments of DNA that are important. And this DNA information is very common now with modern sequencing. They sequence DNA from species all the time, right? Now the idea here is that you're going to compare your sequences for the groups you're studying with an outgroup sequence. So we're looking at a sequence of DNA that's found in a similar part of their genome that the sequence of DNA is probably related to some specific kind of phenotypic characteristic that we observe, for example, and then you compare it. And then the idea here again is to apply the principle of parsimony, so where what would require the least amount of character evolution, or least amount of character changes to get you your hypothesized phylogeny. So here's a case here where you have an outgroup, a hypothetical outgroup. These are hypothetical trees. And here we have a segment of DNA that's only 10 bases long. Remember the adenine, thymine, guanine, cytosine that are the letters of the genetic code in your DNA molecule. So they have this sequence here, and that's your ancestral sequence, your outgroup. And then you look and see what kind of changes have occurred at those specific locations, from location 1 all the way to location 10. And so you can see here, if we look at species D, for example, species D is the only one where T in the 10th position on that sequence changed. to G there, and all the other ones still have just T. Whereas over here, if we look at number 2, it looks like T in that position is changed to C in all of those. So if I had to guess, I would say perhaps maybe this change could have occurred early on there at position number 2. When you do this analysis, though, again, you're looking for the situation in which you get the least amount of change. And this includes careful consideration of homoplasies, right? You don't want homoplasies to mistakenly put two species together that don't belong together because they're actually homoplastic, and that means they would go on separate branches, right? And so we can see that here in this simple example using the same data to construct this diagram. As I predicted from that data set, I said at position 2, all of them changed from thymine to cytosine at position 2 with that point mutation. And that may have happened very early on. Remember, as you go down the diagram, that's further back in time. And that occurred right here at this common ancestor and then gave rise to everything above that. So this would be your shared derived characteristic. Okay. And then without having to analyze the rest of it here, basically this hypothesis would show the least amount of changes that would have to occur in that data set we saw in the prior table. I want to note here that we do have some homoplastic or homoplasies. Remember, these are going to be character changes that we might confuse and think they go together. So without careful analysis, we might put species B closely related to species A because both have that same change at position 8. But it turns out that that change was inherited from different ancestry. Right after we diverged here, way back at this point, this ancestor, we saw another change occur here at this position, okay, as time is progressing. Over here, the change doesn't occur until later and independent of this branch over here. We have that same change here. That makes these two changes homoplastic again. And separating them on different branches gives us the simpler explanation with the least number of changes than if we had to put them together. If we have to put them together, then we're going to see that it doesn't follow parsimony. So there are some issues with this analysis, and they involve the rate at which characters change. And without going into too much detail, because it's not explained well to begin with, it's really beyond the scope of just basic understanding here, is that if the rate at which these characters change is a lot faster, or happens at variable speed over millions of years of evolutionary time, okay, and the possibility for homoplasies may occur, especially when you're analyzing DNA, and all of these can lead to inaccurate phylogenies. Okay, so that's that last point down there. The book discusses this just a little bit. You might read up on it, but don't invest too much time. It's really kind of beyond the scope of what you're going to be asked to know for this course. Anyhow, there are other approaches. There's a statistical approach that you could use, and again, the explanation is very brief and doesn't really facilitate any kind of understanding for us, and it's beyond the scope of this. There are advanced classes of systematics you could take. An interesting thing is that the use of a molecular clock, which is the idea that DNA can change or goes through changes at a regular rate. So many mutations don't occur over so many millions of years that when you do compare DNA between when you can get it from, say, from modern organisms that are alive today, and you compare it, you can kind of give an estimate of just how long ago the groups diverged from each other back on that evolutionary tree or on that cladogram that we were talking about. So when we're talking about normal cladistical methods without techniques like a molecular clock, all we know is the relative amounts of time that those changes occurred. Unless you have a fossil evidence that shows when that divergence occurred. So you see the appearance of these two major groups within the fossil record, and you know the age of the rocks, then you have a better estimate of what time those changes occurred. But at this point, cladistical analysis on its own, without knowing the age of fossils and other things, only will tell you the relative time at which. In other words, the tail evolved long before hair did. Okay, if we're looking at mammals compared to the rest of the vertebrates, right? that's just a relative. We're not saying how long ago that occurred. So that's the idea of a molecular clock, is to actually provide more information on giving actual times in which these changes are occurring. So in this section of the chapter, we are going to be looking at how we classify or organize all of the biodiversity that science has discovered. And classifying, there's a connection, or hopefully a connection between that classification and the systematics we've been talking about. Basically, the connection between classifying and how they're related, these organisms are related to each other evolutionary, evolution-wise, right? So this is really important idea here, because we're going to be looking at biodiversity and doing a survey of some of the biodiversity that we see here on planet Earth. And we have to know about that classification, and then how does that relate to their evolution that got us this biodiversity? What are the relationships? So you're going to be able to explain that classification system. The science is called taxonomy, so it's a taxonomic classification system. And when we do this, we're going to find that these terms that connect the taxonomy to the systematics apply. Some groups that we classify in are going to be monophyletic, some will be paraphyletic, and some will be polyphyletic. And just in there, the root is phylogeny, or evolutionary history, right? So monophyletic would represent a monogroup, a group that shares that one ancestor, right? Poly would be, well, maybe different ancestry in there. So you get kind of the idea of where those terms are coming from. And then we're going to examine a couple of definitions of species and how they relate to this classification. And one concept that's a little bit different in our traditional definition of species is called the phylogenetic species. And every definition for species has its own problems and can be controversial. When I say controversial, I mean in healthy debate within the scientific community on how we define a species. So the science we're going to be looking at is called taxonomy. It is not the same as systematics. Systematics studies the evolutionary relationships. Taxonomy is just simply classifying things. And ideally, we would classify them based on similarities, and those similarities would reflect phylogenies, right, overall. So we're going to be classifying things. Now, the classification is going to follow a scheme, and it's a hierarchy. We're going to call that the taxonomic hierarchy. And that means we're going to be putting organisms into groups that can be nested within larger, broader groups. Okay, so in some ways, this classification scheme may at times reflect their phylogenies, and other times it doesn't. And that's where we have to make the connections between that. All organisms, once they are discovered and described, are going to be given a scientific name. Now, I'm going to say that this definition here for how we name a species is a bit off, so we'll correct that overall. When you identify a species, you're going to give it a binomial. That's two names, and the first name is called a genus. Okay, the second name is not called the species. The species is given two names. It's given a genus name. It's basically classified into a genus, and then that gives it a name. The second name is not called the species. It's called the specific epithet. And the epithet, the specific epithet, is an identifier that is for that species, right? So the second name is not called a species. A species has two names to it. So the species is both genus and specific epithet, and that's where this last point is a bit off. The second word does refer to the species. It is not the species name, so we don't want to confuse that the way it's worded there. Okay, so it's right. It's just kind of confusing, right? So keep in mind the second word refers to the species. It is not the species name. Okay, the species name is the two names. So, for example, humans are given the name Homo sapiens. And the first one is capitalized, and we underline it or italicize if you're typing on a word processor, like Microsoft Word or any other word processor. And then sapiens, it's not capitalized, but you also underline it. no other names will be above this. This is the lowest rank we're going to be talking about for now, the species. We can put species into higher groups, and those groups are not going to be underlined, but they will be capitalized. Okay, so this is the hierarchy. There's actually one missing here. Domain is the broadest group here, and then kingdoms, and then phyla are put into kingdoms. So phyla would be plural. So many phyla can be put into a kingdom, and then many classes are belong to a phylum, and then several orders into a class, and then many genera, plural is genera, are going to be put not into an order. There is a rank missing here. Make sure you add that. It's the family. Okay. So it's kingdom, phylum, class, order, family, genus, and species, and the broadest actual group is the domain. Okay. So I would like you to know the order of this system here. This is the broadest group, or most inclusive, and this is the least inclusive or most specific. So if I ask a question about the order of these, what we're going to call, these are the taxonomic ranks. Okay, so I'm going to refer to them as taxonomic ranks. Okay, so these taxonomic ranks, if I give them to you and I say put them in order from most inclusive to least inclusive, you would be going down this list. If I were to say rank them from least inclusive to most inclusive, then you would list them and arrange them in the opposite order. Basically, be able to write them out without even being given choices. So know them without being able to look at choices. If you know the order, you can say it: kingdom, phylum, class, order, family, genus, and species. Without looking, you know the order. Don't forget to add family into that group. Now, those are the ranks. The actual names are going to be referred to as taxa. Okay. The actual names. Like if I say mammals, that's a taxon. Taxa is plural, by the way. Taxon would be singular. Okay, so this is plural form, and this is singular form. Taxon, okay, with an X. Okay. So you're going to be learning many taxa, and you should know what rank they're at. Is this taxon a kingdom? Is it a phylum? And also, we're not just going to learn them to memorize them for the sake of it. The idea is to connect to their phylogeny, looking at evolutionary relationships in these organisms, with a full understanding that some names may change as we learn more. Okay, so we look here at an example for a squirrel here, and the squirrel. Go back there. They're at the bottom of the list. That's a species of squirrel. The species scientific name, you can see it has got the binomial, 2 names. And the way they wrote it out right here is italicized. The first name, the genus, is capitalized. The second one is not capitalized, and it's in italics. And it's Scurus carolinensis is the scientific name. Just like ours is Homo sapiens, right? So anyway, this one species of squirrel is going to be grouped together with other members that are in that same genus, Scurus. It's the same first name as you saw down here. There would be other genera, probably, of squirrels. This is one genus of squirrels. So the closely related genera are going to be then placed into a family called Sciridae, and they include things like this flying squirrel here, chipmunks, and squirrels. Squirrels would be all in that group, and they're probably in different genera, different genus names, right? And then squirrels are members or classified in a higher group called the rodents, or the Rodentia formally. So the actual taxon is Rodentia, and that's at the rank of order. Okay? Rodents have hair and produce milk for their young, and they're in the class Mammalia. The rodents have backbones, just like birds and mammals and other mammals and reptiles. We don't see all of them here, but we're going to put those in a subphylum called Vertebrata. Sometimes you see the major ranks broken into smaller ones to better fit and better classify. And then we have the phylum Chordata, and that includes the vertebrates, but even invertebrates, weird things that they share. They're related in some way, Like this sea squirt here, it doesn't look anything like an animal that has a backbone, but it is classified with them, and we'll see why later. And then all of these are, of course, grouped with other animals. So besides the chordates, there are other phyla that are there. There's the jellyfish phylum. They belong to one group. There is the flatworm phylum, these flatworms that exist. There is the phylum for mollusks, like the snail. And they're all animals. And one thing they all have in common is they eat, they ingest their food. Now, all of these, their cells have a nucleus, so they're eukaryotes, and they're in the domain eukarya, along with even simple single-celled organisms like this green algae species here, and fungi, and so on. So they would be in the domain eukarya, the broadest group. The other two domains are prokaryote domains you probably talked about in 1406. So our ideal situation here is when we're classifying is that our classification scheme actually fits their evolutionary relationships. However, that's not always the case, okay? Especially when this classification first started out. Now, naturalists have been classifying organisms for well over 200 years, okay? So they've been grouping these things at least in a more formal way for since the 1700s. And so these traditional classification schemes, some of those names still hold true today. Others, when you look at them, you say, how the heck did they group these organisms together? They're not closely related to each other. So here is where those terms monophyletic, paraphyletic, polyphyletic come in. So we're now going to try to connect our classification to the systematics, to the evolutionary history. To give you an example of how names that we still use today might not fit the evolutionary history, just look at birds. Birds are in a class called aves, okay? All on their own group. And dinosaurs are in a group, they're extinct right now. They are extinct, but they're in a group called reptilia. They're with the reptiles. Lizards and snakes and crocodiles, right? It turns out that birds share common ancestry with a group of dinosaurs. So birds would be better classified with reptiles. Birds are basically descendants of a common ancestor of dinosaurs. Okay? So while we still talk about birds as their own group, they would actually, if we're going to consider their evolutionary history, we would put them with reptiles. So when we talk about aves, it doesn't fit the evolutionary relationship. So we have some terms that are going to be used to help us understand when we talk about a taxon, a formally named group, does that group that we're talking about actually fit the evolutionary history or not? So that's where these terms come in. So if that taxon does fit the evolutionary relationships, then it's referred to as monophyletic, and that group would be referring to a clade. So when I say the class Mammalia, for example, it is a monophyletic group. At least that's what is accepted right now based on the evidence. Okay? That means that all animals that are placed in the group Mammalia that have hair and have mammary glands, it's accepted that they share a common ancestor. That would reflect a clade, an evolutionary unit. So again, let's not confuse. The class Mammalia, that's its name. That's its taxon. This is classification. That classification that we put it in happens to fit its phylogeny. Now we're talking about systematics, the connection between the two. Okay. The other two possibilities when we're talking about a taxa is paraphyletic and polyphyletic. So when we look at paraphyletic and polyphyletic, it might be best to look at the illustrations, but let's go ahead and take a look at the definition. You can come back and look at it again. When we name a group and we're referring to a group of organisms, and it includes a group of organisms that includes a recent common ancestor, but not all the descendants. So not every, so we look at a common ancestor to that group, and then we look at all the groups that come off of that ancestor. If you leave one group out, like we did with birds, for example, then that would be paraphyletic. Okay? Polyphyletic would be where you put just where you have a whole bunch of organisms that are put together but they all don't share a recent common ancestor. They basically evolved from other ancestors. So let's look at some examples here. So looking at birds. If we were to put birds really where they would go phylogenetically, where they would go as far as their evolutionary history, they share a close common ancestor with the same group that velociraptors from, think about Jurassic World, Jurassic Park, and T-Rex, it shares common ancestry with them overall, so we should put them together with that. So, and if we did, we might call that group the Archosaurs. That would be a monophyletic group. However, if we left birds out and we just talked about dinosaurs, we see that all the dinosaurs share common ancestry. We can go all the way back, clear back to here. We say, well, here's the dinosaurs, but when we look, one group that shares that ancestry has been left out. So that makes dinosaurs as a group paraphyletic because we've left out some descendants. The descendants in this case are birds. Okay, so that would be paraphyletic. And so, well, that would be the case there. And then if we went and we just made up a group. I want to classify animals with backbones that can fly under powered flight, not just glide, because there's like squirrels that can glide from one tree to another. They have little fleshy extensions of their skin they can use to glide. But we look at those that can actually use their muscles to power their flight. They include birds and bats. Both have a backbone. They're vertebrates. And we want to put them together into a group, and we're going to call that group the flying vertebrates. That group would be polyphyletic because you can see that flight. And here's where if we were to go back to systematics, flight as a character would be a homoplasy. They don't share that characteristic because of ancestry. And so in this case, we would be polyphyletic because bats come off of this branch of that phylogeny, and birds come off of this branch, this with dinosaurs. So we're talking about a group that includes organisms that share different ancestry. So that would be polyphyletic group. Okay, so it turns out that we're going to see quite often in our study this semester that we're going to see classifications that are not monophyletic, and we will be seeing that, like during the study of, say, mosses or bryophytes, like you see in the diagram here above. Bryophytes are not monophyletic. Okay? And in fact, if we start talking about a group called vascular plants, and we'll talk about vascular plants later, but if we look at a group called vascular plants, we're actually leaving out a simpler plant. These are more advanced plants. These are simpler plants. We're leaving out a group that actually shares a close evolutionary relationship and common ancestry with them. That would make vascular plants, and the organisms we put in there would make it paraphyletic because we've left out the mosses over here. Okay? The same thing we might, so when we're talking about the bryophytes here, we've left out the charophytes over here, which are we traditionally put them as green algae, right? So these names may change because we may agree in the future, let's go ahead and change this name and we come up with a new classification scheme, or we may continue to refer to them as these traditional groups that are not monophyletic. So when be paying close attention when we cover groups, and the textbook, my notes, or this lecture here tells you this is a paraphyletic group, we want to make that connection that we're talking about a classification of these organisms that we're studying together, but it doesn't include all of the descendants of that common ancestor. So this brings up the idea of how we define species, which was the lowest rank we saw in our classification scheme. And there's your traditional biological species concept, and this includes a group of organisms that are, or populations that are capable of interbreeding and producing fertile offspring. And these populations could not breed with other populations. So that's how you define it. So if you have groups of organisms, populations that are capable of interbreeding with each other and making fertile offspring, that's your species, and they're isolated reproductively from other groups. That's your definition, your traditional definition. Another definition that has been brought up, because biological species concept has some issues, and I'll tell you those here in a second. But Another concept for species would be to define a species based on shared derived characters. So when you see a group of organisms where they have enough differences, and those differences, that group shares that difference and is different from other groups, then we would call that species. Okay? So let's take a look here. First of all, when it comes to the biological species concept, there's two major problems. The first one is that not all species reproduce sexually. They're asexual reproduction, like bacteria. They just divide. So if we look at a bacteria like Escherichia coli, which is found in our gut, how do you define that in terms of biological species if there's not a male and a female combining genetic information for sexual reproduction? Okay, so bacteria wouldn't fit this definition, okay? But they would fit the phylogenetic species concept we'll see in a bit here. Okay, so because they share that some common characteristic or derived character. Another one is when we have populations that are allopatric, and that means they don't live close enough to each other where interbreeding is possible. There might be a physical barrier like a mountain range or a large lake or something that's separating two populations that in nature would never interbreed because the individuals can't mix with each other overall. If we put them together, could they produce offspring that would be fertile? So here, an allopatric situation, allopatric means other, and patric like patriot, like of a country or of an area. So they're allopatric. Under normal circumstances, they would never be able to, but what if we were to put them together? Could they do it? And if they could, is allopatry really a reason to not call them separate species? The fact that they live separate from each other. So that creates problems with the definition. So the phylogenetic species concept can solve this problem. Those two problems of allopatry, not living together, being able to interbreed, and asexual reproduction. The problem with phylogenetic species concept is that if we wanted to, we could get nitpicky at what a derived character is and continue to keep splitting a species and splitting and splitting and splitting, and we can go down to the individual if we wanted. So this can be a problem. And the cases highlighted here where they have a cladogram, but this is not of different species. This is of the same species but different populations. Okay, so you can continue cladistical analysis within a species when you have a very broad species living in a broad geographic distribution on the planet. They live over a broad area. There are going to be slight differences, so you start analyzing what are differences between populations and use cladistics again, right? So in this cladogram here, your groups are actually subgroups of the same species. So these are populations, population A, B, C, D, and E. And you can see that species A is more distantly related to species E, whereas E is more closely related to D. In other words, those populations right here, D and E, probably are living in an area more close to each other, whereas A probably lives further away, right? But let's say that the area where population C is living, where they're distributed, and the environment changes significantly just in that area. And that creates rapid change in the characters, like, for example, flower color. Enough rapid change to where if we apply the phylogenetic species concept to this issue here, we might see enough difference and say, look, yellow flower color is a good enough derived character to say this is a separate species. So now we take this out. It's not even the same species, and we just put it on a different branch now and say it's a different species, when actually it shared common ancestry with D, very close common ancestry. So what have we created now? We put C as a different species. It's not even in the same species anymore, and all we have now is population A, B, D, and E in their own species. And so now we have a paraphyletic species, because A, B, D, and E would all share their common ancestry and they're together, but we've left out one of the descendants. That flower actually is descended from that ancestor. So there's a problem with the phylogenetic species concept here. Okay, so again, there is no perfect definition for species, but it doesn't make trying to define species unimportant. We still need to try to define species.